Our recording and introduce our two wonderful presenters for today. So Dr. Chris Coffey is a professor of biostatistics and the director of the Clinical Trials Statistical and Data Management Center at the University of Iowa College of Public Health. And Dr. Will Meurer is a professor of emergency medicine and neurology at the University of Michigan Medical School. And both Chris and Will have been longstanding leaders for the CTMC, and so we're happy to have them here today to deliver their joint talk titled Avoiding Pitfalls in Early Phase Clinical Trials. And Chris is going to start for today. Thanks, Chris.
Sure. Thanks for the introduction, Megan. Can you see my screen? Probably not. I don't think I'm sharing.
No,
Hold on. There. Now you can, right?
it's coming. Now we can.
Okay.
It's not in presenter mode, but we can see it. There you go.
You see the presenter mode now?
Yep, and we see the
Okay.
objective slide.
Okay, good. Yeah, so I think what we're going to talk about here, like it's very hard to tell you what to do to design your trial. It's not easy, but less hard to give you some kind of warnings about how not to move into designing your trial. And so we're going to kind of touch on some common things that tend to come up, both in our experience in the course and in other settings. What I would like to start with here, kind of and I'll give kind of the stats perspective, and will kind of also give somewhat of a stats, but also from the clinical viewpoint. I think the biggest thing to come in is that any clinical trial needs to have one or more primary objectives. That seems somewhat intuitive, but really what that means is what is the question that you're interested in answering in the study? And I think while it sounds intuitive, it's a critical piece, and that any clinical trial that you're doing needs to have the ability to answer some question that gives you information on next steps or whether there should even be next steps. I think fundamentally this is one area where, NIH foundation academic trials differs a lot from industry trials, and that in industry trials, often any single clinical trial is part of a larger program that fits into its specific purpose in the program, and it's kind of clear what that information contributes to the larger picture. We don't structure that in quite the same way in kind of the academic setting, and sometimes it's a little hard to necessarily get that information out of it or to get a clear sense of that. And it's kind of, to simplify it, I refer to this as the so what question, right? So if you do your study, you get an answer, what does that gain you? What are the next steps? Does this go anywhere? If it doesn't go anywhere, then is it really worth the time and effort that was invested to do it? And I think this is a particular thing that comes up in the types of trials that you may be working on in this course when you think about next steps, and that often there's a fine line between what we're doing here and that there's somewhat of a question, but it's also someone needs more experience in order to maybe do what they really want to do, and that's what you're doing here. But it can't go too far away from the question. Otherwise, you're gonna design studies that you can't do. So I think you always want to have a sense of this is what I'd like to do. This is the information that I need to collect in order to answer some question. But also, if I answer this question in the positive or in the negative, where do I go from here? And the last thing I'll emphasize is there should always be the possibility of answering that question in the negative. If your trial cannot fail to tell you to move forward or to tell you, that this is a promising line of research, then you probably haven't designed a good trial. If it's always going to move forward, then really the so what question becomes why do you even need to do this if you've already decided that you're going to move forward regardless? Is it really worth the investment, the time, the resources to collect information to move forward to something you had already decided to do? So that's a very high level statement that should be part of your very early discussions related to your trials and the objectives for your trials. So beyond that, it's also important when you're working this out and deciding your question to be very very quantitative and very detailed in what question you're looking to address. So, for instance, many times someone may come in and say, I'd like to do a clinical trial to determine if treatment A is better than standard of care. That's a very high level objective, but in order to kind of put it into a clinical trial protocol, you need much more than just that. So, for instance, a better question would be in population W. What population are you studying? Treatment A at daily dose X, more efficacious in reducing Z over a period of time T than standard of care. So, what are you studying? What is the dose of what you're studying? What population are you studying it in? What is your endpoint? How are you going to measure whether you've achieved? So what defines better in this sense and over what period of time, and what is your comparator? The other thing that comes up a lot in these early studies is secondary objectives. They're often important because sometimes in earlier studies, it may not be entirely clear what the best endpoint is, but it is also something you want to avoid having so many secondary objectives in here that it essentially becomes what, statisticians might refer to as a fishing expedition, and that you're hoping to find something. Also, your question that you want to address here shouldn't depend too heavily on the secondary objectives. Going back to the point that, the so what question, if you do a clinical trial with a primary endpoint and maybe 10 or 15 secondary endpoints, and your success criteria is that if you see a positive trend on any of the endpoints you've looked at, you're going to go forward. My argument coming back to something like that is you really don't need to do the trial. It doesn't matter if your drug works or doesn't work. You're probably going to have some type of trend on at least one of those endpoints. It's going to go forward. So that would not be necessarily a good approach to that. There are two broad types of secondary objectives, and I think it's important to think carefully ahead of time which ones might make sense. One is looking at the impact on other response variables. This would be one where perhaps there are other ways of measuring effect you would like to assess. The other would be subgroup hypotheses that look at the impact of treatment on the primary endpoint within various subgroups. I would generally say there are always exceptions to every rule we say, but for most of the projects that you would be proposing for this course, the subgroup hypotheses as secondary objectives are probably not appropriate. These you're going to need, they're going to be dramatically underpowered. Even in a phase 3 trial, these are often underpowered somewhat, and you're probably not going to have the power to do those. So I would avoid going down that road as much as possible. From your own selfish sense, one reason that you should avoid a large number of secondary endpoints and secondary objectives is if you have a clinical trial that's funded by NIH, or you would have to register it on clinicaltrials.gov. Even in other settings, you would often have to. And in clinicaltrials.gov, when you register a clinical trial, at the end of the study, you have to upload the findings from all primary and secondary endpoints. So the larger the number of secondary endpoints, the more work you're going to have on the back end of needing to upload that information. So that's also something to keep in mind. Kind of that leads into the next point, that each objective should be linked to a primary endpoint. So what endpoint will you use in order to compare that question, to answer your question? It is something in thinking about, in the FDA language, a good endpoint should be something that matters to a patient. FDA uses the language that an ideal endpoint should measure how a patient feels, functions, or survives. Again, this is another somewhat so what question. So if you see a change, a reduction, an increase on an endpoint, does it have a meaning that would mean something that a patient with a disease would find meaningful? This requires some preliminary knowledge of the endpoint. It requires some level of preliminary data that would be available that would allow you to determine how large of a study, what would the required sample size be in order to address that question. It's important, if you're going to use something for a primary, that the endpoint is well known with the properties of the endpoint, because that's going to be important for determining the sample size. I don't want to dwell on this too much because this could be a discussion in and of itself, and get pretty deep, but there's a difference in the outcome and the endpoint. The outcome is what you're measuring. The endpoint is what you're comparing. So the outcome might be what you're measuring over time, whereas the endpoint might be the change over time, where you're looking for instance, if an outpoint is pain intensity measured by a 10-point visual analog scale, An endpoint might be the change in that pain measure over some period of time. For instance, the example here would be over 90 days. So an endpoint can be an outcome, but an outcome is not always necessarily an endpoint. Both are important to specify when you're working on the properties of your study because your sample size, your ability to answer the question of interest is going to depend on how you defined the endpoint based on the outcome that you're collecting in your study.
Oh, Chris has a question. You
Yeah,
might not be able to see the hand up.
I don't see the hand. What is the question? Let me see.
Oh, Kellen, did you mean to ask a question, Kellen?
Yeah, sorry, can you hear me?
Yes.
Okay, so my question is, what I guess, how do you think about what raises to the level of having joint primary endpoints, especially at this stage where you may not have a huge sample size or you don't have a lot of money? How do you sort of differentiate what is worth raising to that level because it's an important question versus what just can't be done in the trial?
When you say joint binary, you mean as like joint primary endpoints?
Yeah,
Yeah.
sorry.
I think, well, I mean, it's always each one is somewhat specific to that situation. So I think it would depend on that, but in a broader sense, I don't think there's anything that would prevent you from doing it if it was thought to be needed. What you would need to think about is how you would combine the information on those two binary endpoints in order to determine whether this trial succeeded or not. And this is where issues like multiplicity would come into play or other type things. And so one of the important questions that I would typically want to look at in designing a study like that was, let's say that neither endpoint showed any benefit. If you kind of ran that through what you're proposing, or this is where simulation can often help, would it still tell you to go forward? In that case, you probably would need to go back to the drawing board on your design. So, in other words, it's a complicated question, but the short answer is there's nothing wrong with doing it, but you should make sure that in doing it, you're not biasing your study in a way that it's going to tell you there's an effect when there's not an effect. Um, Part of the reason why by stressing this is because there's a long history in lots of areas in clinical trials, but in particular in neurology, where results look promising preclinical, look promising in early phase trials, get to phase 3 and fail. Part of the reason for that is exactly this what we're talking about, that some of the earlier phase studies were perhaps leaning a little too biased towards seeing that something worked, whether it actually worked or did not work. And that's more of the important question. I mean, Kellen, does that answer your question? It's a kind of a vague answer, but it's hard to get too specific.
Yeah, no, definitely. That was helpful. Thank you.
Sure. Thank you. This is in the notes. I think these will be made available to you too. These are just some examples of outcomes and endpoints, in cardiovascular stroke, Parkinson's, and ALS. In stroke in particular, your outcome might be the occurrence of stroke, but you have to define how you're going to quantify that. So what you might be looking at is a reduction in the proportion of patients with a stroke over 3 years. In other cases, I've got an example here of Parkinson's with the MDS-UPDRS change over a 1 year period. This is just some examples of what I was explaining on the prior slide. It's also important to think about in choosing the endpoint that your observed endpoint is a function of the true value, which is what you're mostly interested in, but also two other components. Random error, which is something that just exists with the endpoint that you want to try to control as much as possible, but you can never fully control. And then systematic error or bias. And bias is something that we definitely want to try to minimize in a clinical trial. If many of you have been involved in a trial as a site investigator or during your training were involved, one of the big ways that we try to minimize systematic error or bias with our endpoints is by training investigators. Often, if there's some endpoint that requires some amount of subjective testing, you might have to pass some level of certification on the endpoint before, where the intent is to try to make sure if I'm doing a study with multiple sites that there is some level of consistency in how that is applied across sites so that there's not differences in how different investigators have addressed that endpoint. For random error, You want to try to minimize it by just doing what you can in some of these situations, but you're not going to completely eliminate it. And one of the things that is helpful to know coming into a clinical trial is kind of what level of random error to expect with your endpoint. And there are kind of two broad things that you would want to be interested in. I mean, there are many things, but these are two of the most important, which hopefully you've heard before. But reliability or the precision, and then validity. Does it intend what you intend to measure? And so you've got this, schematic here in this figure, which gives some examples that reliability and validity are not the same thing. You can be reliable, like on the left, that would be a situation where the that endpoint would be very reliable in that you're getting a constant measurement under different conditions, but it's not valid. It's far away from the target in the center. The second one to the left would be something that's valid, but not reliable. On average, it's around the center, but it's all over the place in terms of any individual occurrence. The one on the right would be the ideal situation where it's reliable and valid. You're getting something consistently near the middle. And different endpoints may fall different places on this scale. There's also often complications where the endpoints may be. Sorry, was there a question? I'll go. So there are also situations where the endpoint may be evolving over time in the study, where sometimes you're trying to do your best based on what you can do to come up with the best guess to do that. In terms of potential pitfalls, these are a few that come up, I think, most often in what I've seen. There are others that would come in, but one, choosing an endpoint with little known information about its validity, reliability, and ability to detect change. If you're putting all your eggs in a basket of an endpoint that very little is known, it's going to be hard to know whether the properties of that endpoint are such that you can actually answer the question that you have. Related to that, using a new endpoint without proper pilot testing, this comes up more than I would expect, but often people have proposed studies to me in designing where they want to both try to validate a new endpoint in the same trial where they're testing the effect of an intervention. And that is, in my opinion, a horrible idea because one, you can't really validate an endpoint in a clinical trial where especially if you're randomizing to a treatment and a control, because you have some effective treatment that's going to make it hard to know whether, you're kind of in this catch-22. If the treatment doesn't work, yes, you could, but hopefully that's not why you're doing the study. If the treatment does work, then you're not really measuring the relationship between the endpoint and what you're trying to assess in terms of the properties, because you have the confounding effect of the treatment effect should change the endpoint. This is another one that comes up a lot that is misunderstood by many people, but using an endpoint differently than recommended. And a simple example of this would be if there is an assessment measure that is validated in your field, you're going to use it in your study, but you perhaps think two of the questions aren't relevant to your population, so you leave those two questions out. If you change any aspect of a validated instrument, it is no longer a validated instrument. So if you have a 20-item questionnaire that is validated in your disease area, you leave two questions out and do 18, even though it is structured of those 18 questions the same in the validated instrument, it is no longer considered a validated instrument. You would have to go to additional steps to show that that reduced set of questions is valid in the same way that the full set of questions is. So that's even leaving one question out, you run into the same potential problem. So that's something to definitely keep in mind if you might be in a situation where that is something that you're thinking about. A lot of the studies here may be smaller phase, but most of you are probably working in what I would call the phase 2 stage, where and this is, again, kind of my opinion, but my strong opinion personally is that phase 2 are some of the hardest to design. Because in phase 2, you're looking at proof of concept. It's kind of a preliminary assessment of efficacy, and you're also looking to further examine toxicity. What we're in essence trying to do is balance the risk-benefit association between the treatment to determine whether it should go forward. Because you're not trying to, rule out safety concerns, because you can only assess high-level things, you're not trying to prove efficacy. It makes it somewhat difficult because you're not really powered to address either one of those, but you want to have some type of gateway, some type of threshold to determine whether or not you should go forward. And this is where we run into what we typically refer to in the design criteria as go, no-go criteria. And in essence, here, justification of the sample size in the types of studies that many of you might be doing here is not necessarily to show efficacy. It would be to have a high probability of meeting your go criteria if the intervention works, and a high probability of meeting the no-go criteria if your intervention does not work. And so really that's how you would want to justify that your trial is designed and set up and funded in a way that you can address a priori specified go, no-go criteria to get a clear sense of high probability it would move forward if you should be moving forward, and a high probability of not moving forward if you should not. There are a number of different phase 2 clinical trials that can do that. There's not really sufficient time to go through each of these designs. We'll get deeper into some of these in the in-person course. They kind of fall into two broad categories in terms of types of phase 2 clinical trials, some that use different endpoints than the phase 3 endpoint, and others that use the same type of phase 3 endpoints, but they're not powered for phase 3 efficacy. They're powered for other types of go, no-go criteria. I would say, coming back to kind of pitfalls, common errors, I've already mentioned this once, but optimistically interpreted proof of concept studies. We talk about a principle called clinical equipoise in clinical trials that you shouldn't be doing a trial unless there's some degree of equipoise between the two groups. The individuals doing that study, most of you who have proposed a trial, you are likely not in equipoise. You wouldn't be investing your time and energy in terms of designing a study for an intervention if you didn't feel like there was a chance that it worked. What we're talking about with clinical equipoise is more at a higher level in your field. Is there uncertainty of whether this works? Another common error is insufficient exploration of dose regimen and route of administration. There's a rush often to get to phase 3, and perhaps you choose a dose for convenience. It doesn't seem to be a problem. You take it to phase 3. There are many times a treatment may fail in phase 3, and then the concern after the fact is that maybe the dose that was taken forward was incorrect. So you don't want to go too slowly, but you don't want to go too fast. And one of the questions to think about in deciding, are you ready for phase 3, is if the phase 3 failed, would you be convinced that the intervention did not work? If not, why? And if there are reasons why, can you plug those in? Proceeding to phase 3 without adequate early phase data to guide study design. That kind of goes back to what I said. This last one is a biggie. Especially in NIH-funded trials, because I have said on numerous study sections, I'm sure many of the other faculty have, but if a phase 2 trial comes in that looks like the phase 3 study that should be done, but is just a smaller version of the phase 3 study with no differentiation other than we just need to do this to get treatment effects or to decide how to power the study. Any time a phase 2 study looks like an underpowered phase 3 study, in my experience, you could just suck the air out of the room during grant review. Those end up getting absolutely butchered and have very little chance, if any, of moving forward and getting funded. So definitely avoid that. Think carefully about what the go, no-go criteria. I think I've said all of this with the budgetary constraints, so I'm just going to skip forward to the last point here before I'll turn it over to Will. All of this leads to what is the sample size for the study? Sample size calculation is an essential part of study planning. It's something that you will be thinking a lot about as you flesh out your protocol. You want to use this so that you can answer the question of interest. The complicating thing about sample size calculation is it is as much an art as it is a science. And it's a chicken and egg situation in that if you knew everything you needed to do a proper sample size calculation, you would not need to do the trial in the first place because you would already know the answer. And so a lot of assumptions go into sample size calculation. This is where the more information you have to justify your assumptions, the better reception you're going to get for your plan. It's something where one of my pet peeves as statisticians is many of you as clinicians may have taken a statistics course where you were exposed to sample size, but it was mostly through formula. And if you assume this effect, you assume this variance, what sample size do you need? That's the easy part of sample size. The hard part is figuring out how those numbers you plug into the formula, where they come from, what they should be, looking at the literature, looking at preliminary data, looking at other characteristics that you're going to put into the calculation, and what range of conditions. It's very rare that we would use a single set of conditions for a sample size for a clinical trial. We often would look at a range of conditions because we want to show, and what you should want to show, especially if you're trying to get funding for your trial, is that there is some uncertainty in the assumptions, but across a reasonable range of those assumptions, regardless of where it falls, you have some reasonable ability to answer your question of interest in the positive or the negative. So I will pause there and turn it over to Will unless there are any pressing questions before we go forward. All right, Will.
All right. All right. Hopefully you can see my screen. I did that right.
Looks good.
All right, perfect. All right, I will try to kind of gloss over things that I think Chris covered excellently, and I think Chris covered a lot of things excellently. Um, Here's some of our funding for the course, and I also thank AAN and NINDS for this. These are some things that I have funding from, and I think Chris was sort of drilling this in. What's the purpose of clinical trials? Well, they're to answer a question. What are the goals of us as trialists? And what are you going to get out of a trial? But the idea here is to think about where you're going and then update your path based on data that you collect. I think that's a key thing, both for your career, but also for how you approach a clinical trial. Um, so, I'll pause here at each section header. And again, if you put things into the chat, like Dr. O'Brien already did, when is the right time to use a tertiary or exploratory endpoint? There isn't pilot data to support it. I think we could probably address that.
I'm answering that in the
Yeah.
chat. Well, if you
Yeah.
want to.
Oh, okay. Then I'll just keep going. All right. All right. So Pitfalls. I used to play Atari when I was a kid, and this was a game that I loved. It was called Pitfall, and then Clutching My Pearls. So I use this iconography to sort of illustrate whether you're getting a pearl or a pitfall. All right. So when we get through the next 20 minutes, so there's some time to talk, where will you be? Um, Getting the question right is essential. It takes a lot of thinking. It takes a lot of time. Um, but you also want to be wary of myths that people say, like your first trial must always be focusing on safety. If you're testing vitamin C in stroke, showing that vitamin C is safe in stroke when it's like, drinking orange juice, so to speak, it's not really the first step. But the other thing to do is you fast forward to the end of this trial that you're thinking about doing and imagine what it says. Is it an underpowered efficacy trial? As Chris said, you always have to have an option to pivot or stop. Your trial must have a way of telling you, do something else. If it doesn't, then what are you learning? So part of this is then you're sort of creating a vision of greatness. And greatness sounds a little narcissistic, but if people have been in Ann Arbor for a while, I know some of you have. Zingerman's is a famous deli here, and Ari is the founder of this, but really does a lot of continuing education and career development things that describe the power of visioning. So what is it that you are, and I think this is applicable to clinical trials. Like what is it that you're looking to do and thinking it through. So one aspect of it is what information will the trial provide? How does this move the scientific field forward? I think this is also something that when you write an NIH style grant, this is what significance is. Did you do something that moved the scientific field forward? A lot of times in medicine and epidemiology and science, words have meanings that aren't exactly the meanings that you might think of them. Significance isn't really gosh, stroke's bad. A lot of people do badly from stroke. That's significant. It's like, well, no, we lack effective treatments for this subset of stroke patients. That's important. We will learn this about that subset and how to treat them better. That is significant. In addition, how will doing a trial help you move along in your career? If your goal is to discover things that can help patients of the future, then if you're going to take the time and effort to do clinical trials, you're going to want to learn how to do them as well as possible so that you can accomplish that goal of helping the patients of the future, or at least helping the trialists of the future know that they shouldn't do a trial because you already did it and it didn't provide a path forward for a specific disease. And so those are the things you're thinking about. What is it that this trial can do for you? What is it that that you can learn? So will the data change your future actions? This works on both tracks. If the answer is no, reassess the design. And this is both for how you're setting out to do clinical research, but also how you are doing a trial. All right, so what's the scope of the problem? Clinical trials kind of stink. Depending on your climate, you may be familiar with this. This is a stink bug. There are a lot of potential problems with clinical trials, but one way of looking at a clinical trial is you got to make sure it's not, I know the answer, I just got to do some science things to convince other people that this is the answer. That's the wrong way of approaching it. It's more like a diagnostic test. And for those of us who are clinicians, we use lots of diagnostic tests. But if you think about it, power is like the sensitivity of a diagnostic test, and the type 1 error, the alpha, is like the 1 minus the specificity or the false positive rate. So this is what we are doing is we are looking across the sort of universe of potential treatments and trying to determine which of the treatments are potentially efficacious. That's why we want to do a clinical trial. It's to get at this question. But we gotta make a ton of assumptions, like, oh, we had these animal models and the dose was good. Oh, there's not heterogeneity of effect. Ooh, subgroups will respond equally. Oh, let's not do this subgroup. These people are too mild, they're going to do great. Or these people are too bad, they're going to do terrible no matter what we do. We want an effect size, but it turns out we feel like most acute stroke studies are somewhere around 800 to 1,500 patients because that's what the government seems to pay for. But would you be happy to find a thrombolytic that would be, if TNK is helping 50% of the patients and you're treating tens of thousands of patients a year, a new treatment that could help 52% of the patients would be highly helpful, but it would require a really, really large clinical trial. So it's not to say that that's not a meaningful difference. It's just a matter of how one judges whether the resources to do that and answer that question are reasonable. But the opposite isn't true. You shouldn't say, well, I'm going to look for a new thrombolytic, and if it's not 10% better than TNK, it's useless. And that gives you that trial in that sort of 800 to 1,500 range. That is often what we do, but it means that you might discard some treatments that actually would be pretty useful. So sort of measuring what a clinically meaningful effect is versus what would be practical based on the resources that could be possibly asked means there may be some questions that you shouldn't ask or you have to go about in a different way. Noise and outcomes. Chris was talking about the sources of variability, whether it's from systematic bias or random variation. That's very important. I think if you're thinking of it as mortality as an outcome, there's noise in mortality, right? Sometimes people get hit by a bus or die of a different thing, but in terms of your clinical trial, they're counted as a death. Duration of the treatment's practical. Sometimes you might want to give a treatment for a longer time, and this is a little bit more relevant for acute studies. Obviously, in prevention studies, people do take treatments for a very long time, but this is a lot of stuff, right? And can you actually learn about all these things in an exploratory phase trial? No, but you have to figure out where the best places to look are. So what kinds of questions are good? And Chris went through this in a lot of ways, but you should have one main question, and depending on where you are, is X better than Y? Or put more specifically, does X improve a patient-oriented unoriented outcome compared to Y when administered to this population for this dose at this long. So now, can you make the question better, right? And what's the dose schedule and population that would hold the most promise for determining that treatment X can be proven better than treatment Y in a future trial of size this? So that's a different way of looking at it, but it could be a good question. It may also be infeasible. There may be too much uncertainty to answer a question like that, but it could be a good question. And what if you ask the wrong question? I think what the FDA worries about, and I think what us as academics worry about, is maybe falsely declaring an ineffective drug efficacious. So that's how a lot of things are set up. But in that example of, we found a new thrombolytic to replace TNK and TPA, but we only did an 800 patient study, so we threw it away because it was, because it wasn't, there was no way we're going to detect that sort of treatment effect, because it's much harder when something works to find something that works better. Drug treatment trials that are versus placebo, again, are going to be scientifically easier because you're not going to have to incrementally improve upon something that is actually already working along the same biologic mechanism. So if you do a clinical trial and you get something pretty wrong, like, oh gosh, I'm studying progesterone and traumatic brain injury, and I only treated people for 3 days, but I needed to treat them for 7. You do that, nobody's going to go back to look for that treatment, and you might have messed up, right? It might have been that if you had started your first study and treated for 7 days, the study would have been positive, but you won't be able to go back and ask that question again in all likelihood because people will be like, well, if it didn't work with 3 days, what are the chances it will work for 7? You don't know. So that's something that you should worry about. Like if you're doing the study, are you potentially poisoning the well for future treatments? So trying to leave, that's a regret that we should think about it and say, we definitely need to do a study that wouldn't leave us wanting, so to speak. So defining the outcome for these early phase studies is hard. In terms of a lot of times there is a reference outcome that's good for phase 3 studies, but what you're using in your early phase study can be challenging. Chris talked a lot about validation and reliability of other outcome measures. I think there's some questions as to whether consideration of co-primary endpoints should be good. And that's, I think, probably too detailed of a discussion for this talk. But I think it's definitely something that we could think about if it's something that you're thinking about for your specific study design. Knowing the disease phenotype can be important, and this owes to if you pick a really narrow phenotype, you might not have very many patients. So that can lead to problems later. You might have multiple phase 2 trials of the same agent in different diseases going, or you might be able to wrap that into something called an indication finding trial, where you're looking at those multiple different related diseases or disease phenotypes, and then the output is this is most promising in these specific phenotypes. What's the population that should be enrolled? This is a challenge. I sometimes use a lot of stroke examples. If you narrow the population, you'll have less variability. Again, we worry that on the low end of severity, people will all do pretty well. You won't be able to detect a treatment effect, sort of a ceiling effect. And on the high end of severity, there'll be sort of a floor effect because lots of people will do pretty terrible. So you look for this middle space, but then you might be constraining your population too much. And you might be withholding treatments in the future from some patients who would have benefited. I think if you are interested in further I think how we've gone about finding the right population to do thrombectomies in stroke has been a good illustration of that problem and a motivational example. If you go with too broad of a population, clearly there's increased variability and confounding factors. There may be worries about heterogeneity of effect. And sometimes we get into a conflation space where people get a little worried about, maybe we should be doing more effectiveness. And if we narrow down our population too much, we're moving away from effectiveness. We're not creating a generalizable treatment. But the flip side of that is if you can't find a constrained population that will benefit from the treatment, trying to broaden it to a more broad population is probably pointless. So you know, sometimes we get into that conflation of saying, oh, don't do an efficacy study, do an effectiveness study, even when you haven't identified that a treatment is efficacious. You know, one example of that, again, moving back to the sort of stroke reperfusion space, you could consider the idea that you know, when we did the IMS-3 trial, we used a lot of different devices in a very broad group of patients with stroke, including some who did not even have a large vessel occlusion. And because we were using such a broad population, and we were using different devices that were sort of mutating over time. The overall study was neutral because there were so many things that were confounding this, and there were some people who probably biologically weren't conferring any benefit because we didn't even know that they had occlusions. You know, fast forward to the first of the real successful thrombectomy trials, Mr. Clean, and that focused entirely on people who had a large vessel inclusion. You know, the treatment was randomized kind of sequential to intravenous thrombolysis, and they used newer devices that were faster and more safe and easier to deploy and quicker. And it ended up really changing the paradigm in terms of it went from something that looked very neutral because of the way that things were designed and how we were testing this broad population, but then it really transformed into a transformative treatment. But then we also had issues in terms of patient selection of, you know, some vessels are too distal and so forth, and we're continuing to work those things out. But it's important to get that population sort of in this Goldilocks space of just right. You know, it's great if they can have a good, you know, important movement of the treatment in a subset, and then you can perhaps branch out from there to find patients who could benefit. I think in the space of critical care, whether it's post-cardiac arrest encephalopathy or sepsis, you know, one of the issues has been there's a lot of phenotypes in those diseases, but we've been doing large kind of two-arm or sometimes even 20-arm trials in those spaces where we aren't showing a lot of difference between two groups because there's so many other things that are going on in those patients in terms of their critical care they're getting. In addition, though, sometimes the treatments don't end up being too much different, like in terms of sepsis strategy studies. I know this is a neurology course, but I'm an emergency physician, so I care about sepsis. But I think moving away in critical care studies from sort of large, simple trials to trials that more target on a specific phenotype, I think can be helpful. And I think many of the folks in this room who are focused on neurology trials, I think we're into that. You know, there can be other aspects of that though that are maybe less optimal in terms of you know where somebody is on a specific slope of progression for a neurodegenerative disease and testing treatments that only work in that sort of narrow space. You know, sometimes it can be um, thought of a little bit like, you know, is this cheating? But again, if you have a biologic, you know, going back to the thrombectomy example, makes sense to focus on people who have the largest large vessel occlusions, right? Opening them up should make the biggest difference. So when the studies altered and narrowed in on that population, they were able to show a really large treatment effect. There were other populations that we could expand to later, and we continue to work on that, including people in our course who are working on what to do in medium vessel occlusions. Um, but if you don't show that it works somewhere first, you know, it's harder to use that as a beachhead to expand the treatment indications. So I think I'm wrapping up. I will say one other thing that can be very helpful as you try to design a clinical trial is to look at the consort extensions. These are typically thought of as reporting guidelines, but if you're designing a clinical trial, you might want to think about all the things that a good journal like the New England Journal is going to want to see in your report at the end, and make sure you've built those elements into the study in advance. So there are extensions to Consort. There's now been a 2025 update to Consort. So there are a few areas where there's sort of incongruities. There's a 2025 Consort statement, but there's not a 2025 extension for adaptive designs, at least as of yet. So, but these things, if you're looking to do a non-pharmacologic intervention, you're looking to do early phase dose finding studies, pilot and feasibility studies, and of one trials, there's really, looking at the explanation, elaboration, and the checklists can give you a sense of some of the design elements that you should be trying to include. So with that, I think the key takeaways, again, like Chris said, getting the question right is essential. Number two, beware of myths of like, you must do the safety study first. Again, if the safety study is helpful and is going to be informative, great. But if you're doing a thrombolytic study and you want to see what the SIH rate is, and you're going to test it in 10 patients when you're expecting symptomatic intracranial hemorrhage to be seen in about 6%, finding that you only saw 0, 1, or 2 SICH in a study of 10 is not particularly informative. So again, thinking about what it is that you could learn and how it's going to change what you do next. Now, that's not to say that you might say that I can only do 10 patients for a variety of reasons. We're testing other proof of concept things, and we set a semi-quantitative rule of if we see 3 symptomatic ICHs within our group of 10, that's probably so far away from the 6% rate that it is, clearly something is going on there. So again, won't say that, there may not be ways to do it that way, but again, having that be your main study, just understand you're not using, you're really susceptible to random variation there when the numbers are so low. Again, fast forwarding to the end of the trial and saying, like, what did this tell me? But think of game out both situations, and there's even more than 2, obviously, but the situation of where you've achieved your primary objective and when you haven't, and then what would you do next in those cases? You always ask yourself if it's an underpowered efficacy trial. And pivot, doing something different or changing who you're doing it to and when or how is always got to be an option. So with that, I think that's my last slide. So I will stop sharing. And I did finish exactly by 12:50,
Nice.
as I predicted.
While we wait for questions, I did want to expand on one point Will had on his last, because I think it is something that could be useful students of the course. Will mentioned you don't always have to start with safety, but even if you start with safety, I think one other important component, if like safety is the main objective of your study, this is another one of my maybe pet peeves or soapbox moments. Your objective should not be to show that your intervention or treatment is safe. You will never do that, even in a phase 3 trial. All you can really do as you move through the process is you roll out different levels of unsafe. So, usually what you're looking for in one of these early phase safety studies is similar to what Will mentioned with SCH, but you're looking at would you stop if the safety rate is so high that it seems like this should not go forward, which is probably well above the risk of adverse events you would actually be willing to accept in the study. But that's all you can really hope to do in these smaller studies. It's somewhat semantics, but if any of you have safety as a component, that's something to be careful to watch out for, that you will not show that your intervention. In phase 3 clinical trials, we don't show interventions are safe. That's why there's post-approval follow-up that is usually required with FDA or other regulatory agency approval.
Right. Like in my sort of 10 patient thrombolytic study example, by having this sort of boundary of saying, oh, if there's more than 2 out of the 10 who have an SICH, then it's highly probable that the SICH rate is higher than what we expect at 6%. But you're ruling out a level of unsafety. But if you see 0, it doesn't mean that the rate is 0. If you see 1, it doesn't mean the rate is 10. So, yeah, I like you can't show safety, but you can show a threshold of unsafety that would give people pause before moving to the next step. Dr. Gutman.
once again, I learned something. I always learn something. How many times have I heard this talk now or a version of this? But there's always I'd really like people to try to ask questions. I mean, one of the things that I think about when you guys were talking was, um, you know, one of the concerns sometimes is that, your patient population is precious and maybe you actually have a very rare population, right? And so you try to jam as many questions to answer into one trial, like this may be your one big chance, and you don't want to waste your patient population. When actually the reality is you may end up wasting your population if you ask too many questions and you end up with no good answers. But I don't know if you want to reflect on that a little bit, because there are some of the folks in our group this year of rare diseases. So
Well, that was somewhat related to the answer I put in the chat about the exploratory endpoints that I understand what you're saying. And in a rare disease, you might want to collect as much because it may be your one shot at doing a clinical trial. But I think you also have to ask yourself, I mean, all endpoints are not equal in terms of what they add to the study, in terms of cost and complexity. If there's an exploratory endpoint that requires an MRI and your scanner is in a different location, the cost and complexity of that are very different than it's something that's a 5-minute questionnaire that you could add and have somebody fill out. But the other side is, and I think this is also where, I mean, this is beyond probably the size and scope of what the students are working on in their projects. But in larger multi-site clinical trials, it's becoming more common to have a patient or a patient advocate on where you could kind of go through all your different assessments for each visit and get a sense of how long you're going to expect the average person to be at that visit. Because if you're piling too much on people, you're going to end up losing people in the study. And then instead of getting more information, you end up getting less information on some of the key stuff, and then you end up frustrated at the end as well. And Will, you want to add anything?
No, I mean, I guess no, that's a great response. I mean, I think the other thing that Lori would say that I would say to Lori's example of some rare diseases, there's the opportunity cost of that trial, right? And I think if it is a small trial, but there isn't a really good natural history study of that disease, you have to think about that as a maybe a dual opportunity that you're going to get a really good natural history cohort. But again, that doesn't mean you can ask people to come to like 8 hours of testing for like medial plantar neuropathy, right? Like, because um, Lori's joking. Liz is chuckling because I have medial plantar neuropathy. I didn't even know that nerve existed. But then the other thing is, though, in more serious rare diseases, the patients have a substantial opportunity cost, right? Like, because they're like, if I'm in your trial, maybe I can't have this other treatment or do this other thing. So that, I think that was sort of where I thought you were going initially. But again, I think, and then the other thing is, if you're doing like a K23, obviously it's really hard to do a clinical trial on a K23. I would say, avoid. But if you are or something like that, you also have to think of what is the value of your data set that you are creating, even if whatever the intervention you're testing is neutral. And if it is, if it is something that it's a disease where you don't have a good scope of how patients progress, in a real better unbiased way, showing that the data set is valuable, even if the intervention is more neutral or less good than affected can be helpful. But yeah, you do have to balance the human factors of it costs a lot of money to do a lot of tests to people, and you got to kind of sort of drill those things down. Did
I
anybody
think
else?
I'll add one more point to that, that I think Will gave an example that is more of the rare situation and like the thrombectomy where there were trials that didn't succeed followed by trials that did. The hard part in many cases is sometimes you just have to face the fact that your intervention doesn't work. It's really hard for people to accept that in many cases. We're
I think
usually the targets. The first time a trial fails to show something, it's because the design was flawed, when probably it's more that there were problems with the intervention or the population. But sometimes they don't work.
yeah, but I think the thing about thrombectomy where there is like it would seem like getting a clot out of an occluded artery ought to work. Like there's like an extreme prior of biological plausibility that a brain with blood flow at some level ought to be better than a brain without blood flow. And I think there had been a lot of controversy in the emergency medicine field about TPA doesn't work for stroke. And I think it may be that in certain patients, certain subpopulations, it may not work as well. The benefit risk ratio is not even for every single person who walks in the door with a stroke. But, I think when a treatment doesn't work, it is under the conditions that it has been tested. This treatment didn't work. But you're right. If it wasn't biologically, if the biological plausibility isn't there, it's hard. But Dr. Aziz.
Yeah, I guess just to go off of this example that you've already raised, kind of trying to find the true phenotype where your intervention is going to have the best effect is I sometimes struggle with the inclusion exclusion criteria, which I'm sure is going to be a talk for another time, but of being too restrictive and what's kind of the sweet spot between making the compromises and making something a little bit more pragmatic as opposed to making sure you're hitting your target demographic for your
I guess I think probably, Chris, I'm sure has some feelings on it too, but I think it depends on the situation. If it's a rare disease and you're, getting things down to an even rarer subset of people with a rare thing, then it's going to be harder. If it's a common but hard to enroll disease because it requires 24/7 enrollment, like something like stroke, again, you're increasing costs by making it harder to figure out if people are eligible. On the other hand, if you're saying, I'm only going to enroll people who are aged 45 to 61 and have an NIH stroke scale of 7 to 10 that includes none of the ataxia points, um, there may be good reasons to do that, but then you've increased the cost of the study and you've increased the length because people are going to have to go there. And then you've also had this issue that the testing of ataxia has a lot of inter-rater variability and is confounded by the degree of motor weakness. So you might not even, and again, that's a really dumb idea. That's a dumb sort of set of inclusion criteria for a stroke trial, but it gets at, some of your exclusion criteria may be more subjective or inclusion may be more subjective or, than others too, which adds an additional sort of layer of trickiness by trying to be too cute in terms of having the population. So I don't have a good answer as to how to drill that population perfectly. But if there's a biological construct to say, people who have medium vessel occlusions who have NIH stroke scales of 1, we're probably going to avoid because they're going to do pretty well. I think there's a good biologic construct for that. As an example.
I think we're at the top of the hour.
We are. Thank you so much, Chris and Will. Great presentation. Thank you, everyone, for joining. You can use this QR code to grab the survey, and we'll hope to see you at the next webinar.
Please do the survey.
All right, bye.
Thank you. Bye.